Check. Can you hear my voice? Are my voice clear?
Yes, I can hear you loud and clear.
No, your sound is not in my head. Can you speak again?
Tes.
Coba.
Hah?
Ngomong.
Ini lagi ngomong.
Thank you. My voice is clear, right?
Yes.
Okay.
Coba Rahmi cek sound.
Oh, kalau itu ya kedengeran Pak.
Kamu nggak pakai ini TWS kan? Nggak ada ya. Suara backgroundnya kedengeran banget atau pindah ruangan aja?
Boleh deh.
Lagi 205 kali.
205 aja. What? Iya.
Suara itunya kalau nggak pakai headset kedengeran banget. Rahmi kayaknya ya. Sama saya. Harusnya dilihat mikro. Nanti diskusi sama Pak Puji deh, biar taruh mana? Nggak mentor di proses. Sama mentor segede gabang, mejanya sekecil. Terus saya juga. Ya fermentornya segede gabang.
Cek.
Ya Pak Puji.
Oke, suaranya udah jelas ya?
Sudah Pak.
Oke, ini apa? Yang anak-anak sudah dikondisikan belum ya untuk segera join?
Tadi saya sudah minta Kak Sekar Pak. Jadi saya minta 2.20 itu semua sudah join. Jadi biar 2.30 pas langsung mulai.
Iya. Nah,
Mungkin
takutnya
beberapa
keburu
menit lagi Pak.
Prof Chongnya join, kita belum.
Iya Pak. Saya ingetin lagi coba Pak.
Iya, coba di itu lagi. Guys, buka dulu. From and also to Allah Subhanahu wa Ta'ala for His blessing that allow us to be here today for this visiting lecture, even in good this visiting lecture of our to enhance academic quality and strengthen international collaboration. Today we are only fortunate to have Prof. Chong, who will deliver a lecture on the topic harnessing biopesticide and beneficial benefic. Yes. Yeah. This topic is highly relevant to the challenge of modern agriculture, particularly in addressing plant disease and the urgent need for environmentally friendly crop protection system. I sincerely hope that both student and lecturer will take the opportunity to learn, engage in discussion, and even initiate potential research collaboration in the future. On behalf of the Vietnam Study Program, I would like to express my deepest appreciation and gratitude to Prof. Chong for his willingness to share his valuable insight with us. Halo, selamat sore, Prof. Chong.
Selamat sore, Pak.
Waduh,
Pak Puji, apa
sampai ketemu,
kabar?
Prof. Iya. Di mana ini, Prof?
Saya di office, Pak.
Oh,
Pak, di
ya,
pejabat
di
office,
pejabat.
office ya.
Oh, office. Enggak, infonya di luar negeri itu kemarin.
Apa
Prof.
ya, Pak?
Chong lagi tidak di Malaysia.
Saya di Malaysia. Saya 18 baru saya
Oh,
pergi,
di Malaysia.
18 Oktober.
Oh,
Tanggal
iya, iya,
18
iya.
Oktober baru
Oke, oke,
pergi
oke.
Asian Agri di Medan. Ah, ah.
Oh, yang 18 itu ya, Oktober ya. Iya,
18
di
Oktober
Medan.
Medan.
Oke, oke. Iya. Ya, sebenarnya kalau bisa offline oke ya, cuma waktunya kita kelewat itu programnya itu.
Oh,
Iya, 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 iya. Gimana
terlewat.
kabarnya, Prof. Chong? Baik-baik saja.
Baik-baik saja, Pak.
Oke. Iya. Ini kemarin mahasiswa dari UMS Sabah juga ada kegiatan dengan kita, apa namanya, menyelenggarakan apa itu kayak sukses story dari alumninya gitu.
Oh, di Ponorogo,
Iya,
di
tapi
Undip.
online juga. Terus ini nanti hari Minggu tanggal 5 ya, tanggal 5 ada lagi itu. Prof.
Oh,
Iya,
tanggal 5.
dokter laki juga ada kegiatan juga.
Oh, oke, oke. Tanggal 5 ya.
Oke. Iya. Wah, itu kangen ingin ke Sabah lagi saya. Oke, mungkin bisa dimulai, Pak Irfan, Bu Nisrina, sudah.
Ya, Pak, baik. Sudah
Oke.
siap. Sebentar lagi akan dimulai oleh MC-nya.
Oke, silakan.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon,
Waalaikumsalam
ladies and
warahmatullahi
gentlemen, respected visitors, and dear students. It's really great honor to welcome all of you guys in today's event. May I introduce myself first? My name is Rami Afifah, a second year biotechnology student at Diponegoro University. And on this fine day, I'll be your master of ceremony for today's event, the visiting professor online lecturer with the team harnessing biopesticides and beneficial microbes for sustainable crop protection of a simple oil from Ganoderma. And all right, to start our agenda, let's listen to the welcoming speech from the head of study program, the head of biotechnology study program, Dr. Sri Pujianto, SSEMSE. Please welcome. The time and place is yours.
Oke, halo, cek. Assalamualaikum.
Waalaikumsalam. Sudah
Oke,
masuk suaranya.
oke. Baik, terima kasih atas waktunya, moderator. Jadi, sebelum kita acara puncak, pemaparan materi oleh Prof. Chong, kami selaku Ketua Prodi S1 Bioteknologi, pertama menyampaikan terima kasih kepada Prof. Chong, from UMS ya, Universitas Malaysia Sabah, atas ketersediaan waktunya untuk menyampaikan visiting lecture by online di tengah kesibukan beliau. Terima kasih Prof atas waktunya. Kemudian pada para mahasiswa, jadi ini program visiting profesor ini diselenggarakan dengan dana WCU ya, World Class University, dari Fakultas Sains and Mathematics ya, tujuannya adalah memberikan pengalaman secara langsung dari ilmuwan-ilmuwan kelas dunia ya. Jadi saya tahu Prof. Chong ini merupakan salah satu the best scientist ya di Malaysia ya. Jadi publikasinya sangat banyak, H-index-nya juga sangat tinggi. Kita akan menimba banyak ilmu dari beliau khususnya dalam apa namanya pengendalian hayati hama pada kelapa sawit ya, oil palm di Malaysia itu beliau adalah pakarnya. Jadi waktu itu saya ketemu Prof. Chong, kemudian beliau banyak sharing bagaimana mengendalikan Ganoderma pada kelapa sawit. Nah, pada kesempatan kali ini kita akan belajar dari beliau. Ya. Saya kira ini merupakan eh, peluang yang amat besar bagi kita juga untuk mengembangkan kelapa sawit di negara kita ya. Baik, semoga nanti kita semua bisa mengambil hikmah dari acara ini. Saya kira itu. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much to Dr. Sri Pujianto for the inspiring words. Okay, next we will have a group conversation. We kindly ask all the participants to turn on your camera and smile and stay still for a few seconds while the committee takes the picture. Okay, I will start with the first page. I will count. Okay, I will count. 1, 2, 3. Okay, second page. 1, 2, 3. Next. 1, 2, 3. And the last one. 1, 2, 3. I'm sorry, the last one. Okay, 1, 2, 3. Okay, thank you everyone. You may turn off your camera again. And now let us move on to the main session. For that, I would like to hand over the event to our moderator, Mr. Ivan Anwar Fauzan, SSEMSE. Please welcome. The time and place is yours.
Okay, is my voice clear, Rahmi?
Yeah, it's clear.
Okay.
Bukan.
Okay. Okay, good afternoon everyone. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Okay, welcome all to welcome to our program, World Class University program that organized by Biotechnology Study Program, Faculty of Science and Mathematics, Benefical University. Okay, let me introduce myself. My name is Irfan Anwar Fauzan. Today I am the moderator in this event, and I am actually a lecturer in Biotechnology Study Program, Diponegoro University. And today we are truly honored to have with us Prof. Chong Kim Pin from Biotechnology Program, Faculty of Science and Mathematics, Faculty of Science and Natural Research, University and Malaysia Sabah or UMS. Okay, maybe I will read the curriculum vitae of our speaker. Please, the operator. Okay, let me introduce our speaker today. Prof. Chong obtained his PhD in plant science from the University of Nottingham, United Kingdom, and his master degree in plant biotechnology from Imperial College London. And he also have so many publications, over 130 research papers and several books, especially in plant biotechnology and agriculture. And he also received Top Research Scientist Malaysia Award from the Academy of Science Malaysia in 2020. And also he served as National Steering Committee member for Malaysia Sustainable Palm Oil. And also he has been invited as several keynote speaker and visiting scientists at several universities across the UK, South Korea, China, India, and also other countries, reflecting his strong international recognition, especially in plant pathology, sustainable agriculture, and also plant biotechnology. Okay, and then today Prof. Chong will deliver a lecture entitled Harnessing Biopesticide and Beneficial Microbe for Sustainable Crop Protection from Oil Palm Ganoderma. And hopefully this lecture will explore how biopest and beneficial microbes can utilize in the sustainable crop protection from oil palm Ganoderma. Okay, mungkin let us welcome our speaker. Hello, Prof. Chong.
Hello, Pak Ivan. Yeah, thank you
Okay,
very much.
okay, thank you very much. Okay, please. I'm actually very curious about your research and the topic that will you bring today. So please, without any further ado, please you can share with us about what you want to share. Thank you.
Okay, yes, please. So I'm going to share my screen. Yeah.
It's still loading.
Can
Okay,
you see my?
yeah,
Can you see the screen? Okay.
okay. Your screen
Let me
is
just enlarge it
displayed.
first.
Okay.
Okay. Okay, now you should be able to see my full screen.
Yes, Prof.
All right. You can see my screen now? Or
Oh, yeah. It's clear. It's clear in the screen. We can see the screen.
the PowerPoint?
No, it's the title page.
Yeah, how about this one? The title page.
Yeah, still the title page.
This one?
Yeah.
It's moving. All right.
It's moving.
Okay, once again, so thank you so much to Pak Puji, Dr. Puji, and also Pak Irfan also for organizing this talk. And I would say this is a sharing, maybe a try within this less than 2 hours time to go through this topic, which is a very topic very close to my heart. The topics given is harnessing biopesticides and beneficial microbes for sustainable crops protections, lessons learned from oil palm Ganoderma. So I hope the sharings from my presentation today will benefited the participants in this talk. So basically, my name is Chong Kim Pin. I'm from Faculty of Science and Technology. There's just a rename of the faculty recently from Faculty of Science and Natural Resources to Faculty of Science and Technology now. Yeah, from University Malaysia Sabah. I think in Indonesia we use Universitas. In Malaysia we use University. Yeah, so just the spelling is slightly different. Yeah. Okay. So I will try to go through this about 5 topics here. The contents of presentation. The first, I will quickly go through a so-called of introductions to biopesticides. So we have a general pictures about the biopesticide first, before we discuss more about Ganoderma, and then which is the pathogens the causal pathogens of the basal stem rot disease, and a bit on oil palm and the disease, and finally we'll go to the sustainable basal stem rot disease management and what actually we learn. We have learned from this disease management and how actually these biopesticides can be further developed. That will wrap up the whole presentations of today lah. Yeah. Okay. So a quick overview about what is a biopesticides. Maybe this will benefit some of if there are some postgraduate students here. So biopesticide actually is nothing new. I mean, it have been promoted. It have been actually introduced long time ago. But of course, if you try to go through, there will be a lot of different terminologies, or how actually people defines biopesticide, and sometimes can be very confusing. Especially in developing countries like us, in Malaysia and Indonesia, and maybe some other developing countries as well. The definitions can be quite confusing. Some people may try to use these definitions, bringing in different type of combinations of microbes, combination of controls, and so on. Yeah, but generally, this is how it looks like. It's actually defined as mass-produced biological control, biologically best agents and used to control plant pests, diseases, and weeds. And it can be very wide spectrums, depends on countries and depends on how actually it's being registered. So it can be, again, I put at the last day, we can see that there may be variation across the countries. But generally, there will be like 3 basic categories, how this biopesticide is being categorized. The first one is, we are talking about products that is actually based on maybe pheromones or maybe semiochemicals. This is mainly used especially in controlling pests, where insect pheromones are used for trapping and for matting disruption. So this is the first category of biopesticide. Using semiochemicals, where chemicals is actually one of the main content or main ingredient to be used in this first category of biopesticide. The second category, this is maybe I will explore more when I discuss about Ganoderma, which is a product that actually very close to our heart, and we have been actually screening. And I will share also some examples that actually we have from Indonesia also, in collaboration, and also some of our experience with our researchers from Indonesia as well. So which is using microorganisms or microbiomes. So either bacterium, fungus, or even virus. And these are the second category. And the last category, which is the category 3, this is actually products based on plant extract. So biopesticides can also come from plant extract. So meaning that we are using some extract from other type of plants and use it to control some of the unwanted pathogens and so on. And this is another next category, which is maybe not depends on country, which actually, for example, genetically modified plants. This one may be not universally accepted as biopesticides, and again, it depends on country and depends on how actually we categorize them. But the common are the 3 categories that we have been discussing earlier. Okay, so we're moving on into the modes of action. So when we try to screen through, we try to develop, or we try to understand about biopesticide. So these are some of the mode of actions that we may need to explore further when I discuss later using some of the examples and so on. So these are things that we may need to look into. The first is whether the biocontrols that we are using are competing in terms of space. This is maybe the most basic or the most conventional biocontrol or biopesticide, which is when we introduce a new biological control agents into the area where our pathogen is, the biocontrol is just competing for the space, meaning that they may have a characteristic that they grow very fast. So meaning that this biocontrol may grow very fast and take the space of that area, that not allowing the so-called pathogens to grow. So meaning that the speed of the growth of this biocontrol should be much faster than the pathogens itself. So this is the first mode of action. The second one is the biocontrol itself may also competing in terms of nutrients. So meaning that they grow fast, they conquer the space, and then they also absorb the nutrient. They're not allowing the pathogens to grow further. The higher levels of biocontrol may be the third, the fourth, and the fifth one, which is the productions of antibiotics. We will see some examples after that when I try to share about Ganoderma experience and so on, where our biocontrol will produce antibiotics that actually directly kill the pathogens that we are aiming for. And other examples of mode of actions also including direct parasitism, meaning that our biocontrol will actually attack the pathogens directly through a direct parasitism. And higher levels and probably a better control, which is actually induced resistance. So induced resistance is the last mode of action we're going to discuss with examples later. When we see the application of this biocontrol is not only providing the other mode of actions, but it's also try to induce, merangsang, induce, so that the plant itself, before the pathogens arrive, the plant is already ready to fight against the pathogen. So this is what actually we say induced resistance. Okay, if we go through in the world market, basically we have a lot of different microbes that are being used for biocontrol, ranging from bacteria, viruses, fungi, and to protozoa. But of course, if you look at the list of market, we notice actually bacteria and fungi took up the biggest market when it comes to biocontrol. Most likely because of it's actually easier to handle, I believe. That's the reasons that people tend to work with bacteria or also fungi when they are trying to develop biocontrol. Okay, I quickly share with you, this is a so-called when we try to screen through developments of a biocontrol product, these are some of the tests that we need to go through. We share the example after this, but this is the general overview first. So from screenings or isolations of so-called candidates ataupun calon-calon yang sesuai. So when we screen through, these are some of the basic process that we need to go through. The first is the identifications and characterizations mode of action, which is the 5 that we were discussing earlier. Let's say if we have a candidates that we think that is possible to be developed as a biocontrol against certain pathogens, the first is we will do an identification and we try to characterize them. So we understand this biocontrol better and how it works. This is where actually the mode of action, whether these potential biological control agents will actually provide a direct parasitism, or is it just competition of space, competition of nutrient, or maybe they are producing antibiotics. Then the second step is we look into the so-called bioassay. So we try to screen through different process. The basic one, of course, is the in vitro. In vitro, again, it very depends on different researcher. Some may say that, okay, in vitro, at least we need to have 80% and above of inhibition before they are claimed to be good enough to proceed further for further investigation. But some may claim maybe like 90 and so on. Then some researchers can go down to 50 or 60. And again, it depends on researchers. But for me, Ganoderma later on I will say that it's 80% at least. Yeah? I will share with you some experience later on. And after going through the in vitro, of course, we will try to use in vivo, in planta, and so on. And only with that, this is involving lab and also involving nursery trial. Then we can proceed further into the so-called pilot test. So we can upscale and so on. And finally, the challenges that we have discussed a little bit also on when we try to scale up. And of course, everything looks good when we try to do it in the lab or we try to do it in the nursery level. But when we try to upscale it and try to come in bigger formulations, it may be so easy to produce 1 liter or 2 liter, for example. But once we start to upscale it to 20 liter, 50 liter, 100 liter, and so on, so the quality will be very different. The number of cell counts, the number of spores or whatever will be very different. So these are the challenges that we need to look through when we develop the whole pipeline from screening until the product itself. And of course, some other tests that may involve, after we have selected a strain, we need to look into the toxicology, meaning that we are not trying to solve a problem. It's actually we are trying to manage a plant disease, for example. So we come with a biocontrol, but we are bringing some other toxicology problem to the environment or to the agriculture products and so on. This is the thing that we don't want. So need to undergo toxicology tests, commercial tests, and of course, we need to patent our formulation. Yeah, and then finally registrations for use and so on. Okay, some of the characteristics of an ideal antagonist for commercial development will be this, meaning that when we go through, so these are some of the criteria that we from our lab look into when we develop our biological control agents. The first is should be genetically stable. So meaning that we don't want it to be keep changing after we have formulated it and so on. Then effective and low concentration. Because from experience, we have actually tried some products which actually effective, but the problem is it is not practical because they need a very high concentration. And applications with a high concentration is not practical when it comes to aesthetics or field which is very far. And not fastidious in its nutrients requirements, ability to survive in an adverse environments and so on. So these are some of the criteria that we did look to look into. Effective if possible. It is effective against a wide range of pathogens. Not only meaning that when we develop these products, it will be good if actually it can be applied to different commodities, not only with one type of commodities. Then the production cost must be inexpensive, easy to dispense. We have experienced actually a product that actually asks us to test. Unfortunately, when we do the testings, it is actually so difficult to dispense. It need to be fermented first for 2 days before we can use it and so on. So, which is actually not practical for the farmers to be used. Then, of course, the product itself shouldn't be detrimental to human health. The formulation shelf-life might be long, resistant to chemicals, and finally, it can, if possible, is compatible to commercial processing procedures and so on. In our lab, we develop our own biocontrol. At the same time, we also help the industry to test the so-called different formulation or products from outside. Okay, so we encounter quite a number of different products that actually very, I would say, very fussy and it's not practical to be used. Where we see, for example, if you want to apply these biological control agents, then we cannot apply fertilizer, we cannot apply pesticide or even other herbicide, for example, at the same time. Or this is going to be very challenging. I mean, when it comes to operational and so on. Okay, the slide is not moving. So, okay. Okay, moving to the second part, now you have a general idea. We are going to discuss about biopesticide. So some of the criteria that we need to look into when we discuss about biopesticide, then some of the board of actions, how they works against these pathogens. Right, let's moving into some of the things that are very close to my heart. I've actually been working with these pathogens for quite some time. I believe Indonesia is also leading when it comes to Ganoderma research. Because the industry of oil palm is very big in Indonesia and also in Malaysia. For the past 20 years, actually, I've been working 20 years working with this disease and pathogen. It's very challenging. We come up with a lot of different detections, early detections, and also management, how to control. But I will say that until now, we still haven't find the so-called of absolute or the best control, I would say. Okay, so if you look into Ganoderma, basically, it's actually referring to a genus, the taxonomy of Ganoderma, and of course, under the kingdom of Eukaryota, the phylum of Basidiomycota, and these are the so-called taxonomy of the Ganoderma. There are over 250 species, or probably more, so many different types of species of Ganoderma. But not many are very popular, but there are only 7 actually we said are very popular species. So if you look at these 7, so basically always being reported is the Ganoderma boninense, the one you can see here. Then the Ganoderma lucidium. This is the one that's the Chinese population use it for medicinal purpose. Yeah, so not really medicinal, they use it, yeah, maybe for supplement to enhance the body. This Ganoderma lucidium. And there are also other Ganoderma like Sinensis and then Zonatum, Apranatum, and also Capensi, and also Suge. So a few different. But the one that actually giving us a big trouble is this one, Ganoderma boninense, yeah, which is the species of boninense. Yeah? So we discussed more about this. So basically, if we look into Basidiomyces, they are filamentous fungi. They reproduce sexually by formation of specialized what could they call club-shaped end cells, which is basidia. So they are it's similar. I mean, among these basidiomycetes. So they specialize spores in this phylum with the name of basidiospores, and they have basidiocarp and so on. So they share some similarities. For example, the presence of clamp connection, which actually we can see this present also in other Ganoderma, not only on boninense. However, if we go through here, as I mentioned, there are about 7 species that are they are more popular compared to the another 250 species. Yeah, so they are actually being reported causing so-called of diseases in different type of crops, even including coconuts and also other crops. For example, we can see this. I just go through some of the so-called of diseases that have caused by these Ganoderma species, but a different species. For example, this is in Acacia. In Indonesia, this is caused by Ganoderma philippi. All right. Then we have Ganoderma planatum, which is killing the aspens in western state of USA. So it's everywhere actually, different species of Ganoderma. But like this one, lucidium, is and Aplanatum also reported to cause problem to winterberry. Okay, so they are there for quite some time and causing problem. And this one again another species. Yeah, so these are some of the examples causing problem in almond trees. Yeah, but in oil palm, we only worry with the Ganoderma boninense, one species only. Of course, there are also Zonatum, which is being reported can cause problem, causing basal stem rot, BSR. However, boninense is still the major concern So there are so many diseases affecting oil palm in other countries in Africa and America. So if we look at a little bit about oil palm, maybe we have quite a number of oil palms in Sumatra especially. So and in Malaysia, basically we have a lot of there are so many different sort of diseases affecting the oil palm. We have vascular wilt, we have bud rot, sudden wilt, red ring, and so on. But these are mostly in Africa and Americas. But in Malaysia and Indonesia, basically the basal stem rot is the major concern. Yeah, Southeast Asia. This is the report actually come from IOPRI and also from MPOB. Indonesia is IOPRI, like Indonesian's Oil Palm Research Institute. Yeah, so the losses actually caused by this basal stem rot disease, which is the pathogen is basically Ganoderma bunians, is about $1 billion. It's not million, it's $1 billion for Indonesia in 1 year. And for Malaysia, it's approximate about $500 million a year. So annual losses is very, very big. That's why my patient with this disease is still there, because for me, this is a disease actually, whoever can solve the problem, you really can make a lot of money. Yeah. So because this is really, if you try to look at the losses 1 year, if you can take you can earn 1% from there, or maybe 0.1%. So it's already a very, very big market. Okay, so this is why I think for the past 20 years, I haven't give up working on this disease. It's very challenging actually. Okay, we quickly go through how it works. This is the life cycles of the Ganoderma. So like some other bacillomycetes, depends on how you look at it. If you start from the spores, so when the spores germinate, this is an N. If it germinated, it become N. Then of course, it can, what do you call this one? Come with the hyphae, and then your myceliums and so on. All right. But interestingly, the singles, the spores that actually, the hyphae or mycelium that germinated from the single spore with N, actually it cannot cause infection. Okay, we call it as a monokaryotic. So it's just disperse, it just grow, for diversity or whatever purpose, but it won't cause infection. But they need to mat first to form the so-called dikaryotic, the 2N. I hope you can see my error. I'm pointing the things. Okay, so 2N, this is the one actually can cause infection. Yeah. And of course, after infecting the oil palm, it actually fit enough, and then they will form again the so-called dense mycelium to form the fruiting body, which is this one, right? The fruiting body, and then again the life cycle go back again in dispersing the spores. Okay, so this is the life cycles of the Ganoderma. And then how does it infect, or how does the Ganoderma spread? Oh, basically there are a few different so-called theory, and some is proven already. For example, the easiest one is actually the so-called root contact. Okay, where the healthy palms contact to the source of inoculum. That's why the sanitation in the infected area is extremely important. So this is what is being done for the time being. Is well practiced in Indonesia and also in Malaysia. And I would say that this is probably the only solution that they have been doing, which is sanitation. They try to make sure that the area they're being infected, they try to remove as much of infected materials as possible, which is very, very costly. Very, very costly. Yeah. So if there is any remaining materials inside there, so once your healthy palms, when you put it there, it start to grow, so that when the roots touch the infected materials, where the stem and so on, then the palm will be infected again. All right. Then the next one is we said that from the crossover between the healthy palm and the infected one, meaning that I'm sure that you have seen oil palm extract before, where you will see that if you want to look to know how far the roots of the oil palm can grow, it's basically like the frond. If the frond is that long, so the roots actually going that far most of the time. So in that case, the chances for the healthy palm's roots to touch the diseased root is very high. So that's why the current practice, they really need to remove the so-called infected palm. Yeah. Another one is the possibilities of spores causing the infection. This one I put some questions mark there, because this is some speculation, and until now we can't really prove it. Actually, this is the so-called of the very high potentials of spreading the disease. Because if you go, I think maybe about in the morning, 8 o'clock, 9 o'clock, or 10 o'clock in the morning, you go into the plantation, you can see the heavily infected areas. If the presence of the fruiting body is there, like this one, yeah, the fruiting body here, you can see that dispersals of the fruiting bodies. It's just like people smoking. Yeah, so smoking, so meaning that there may be millions of spores is being dispersed within that 1 or 2 hours and so on. So these are spores will actually fly everywhere, brought by the winds and so on. So if the spores can cause a direct infection like that, I believe the spread of disease, I mean, may collapse the whole estates already. Okay, so meaning that, but there is a reasons why we are putting that spores still can cause infection, because they may need a very specific condition. I did mention about the monokaryotic and also the dikaryotic, where actually the dikaryotic is the one that actually can cause the infection. So this is the so-called spread of Ganoderma. Okay, so these are some of the symptoms that we probably will see in the plantations or the estates. So, but unfortunately, all the diseases, all the symptoms that you see here, for example, unopened new fronds, wiltings of green fronds, then the fruiting bodies, and this one, the one on the right side, the appearance of fruiting bodies, or even the so-called decaying and so on. This type of symptom we probably can see only after 6 or 7 years. Yeah. Not 6 or 7 weeks or 6 or 7 months. It's about 6 or 7 years, which is very, very slow. Yeah. So that's why sometimes most of the time, the planters actually fail to identify that the palm is being infected. Okay. So to date, there's no any proven fast, low cost, reliable, and practical in situ detection. So the palm may looks healthy, growing like that, but when you see the yellowish or the wilting or something, it's because of the translocation of the water is being disrupted. Okay, in that case, then only you can see this type of symptom. And in some cases, the farmers or the planters may also so-called of mislead because of maybe lack of nutrient, fertilizer is not enough and so on. Yeah. And most of the time, detections are still using a very manual, which actually by observation, look at the symptoms and so on. Even actually, on 18th of October, I'm going to Sumatra Utara for one consultancy work. Actually, we will use our tomography to detect. They are working. This company is working with companies from Japan and also from Malaysia. Also, from Malaysia, we are from Malaysia. So the Japan one actually used the drone to fly on the top of the tree by looking at the canopy, basically the changes of color. But sometimes the color itself from the top can a bit misleading, yeah, because the yellowish can be lead to many aspects. It can be because of lack of nutrient, nutrition deficiency. It can be because of water stress and so on. So our team actually will use the tomography method, which actually will put in the sensors to the palm to generate the image of the infection and so on. So there's no the market industry is still looking for a so-called in situ detection. In situ is meaning that they can immediately check the palm in the field and can confirm that this palm is infected, yeah, without any symptoms. So this is what they're looking for. Most of the time, the detections now is if the palm without any symptoms, although with infection, they may only need to bring back the tissues to the lab and so on, which is not that practical. And there's no absolute control of the disease, although there are some promising results now. Okay, we are also reporting the results, and of course a lot of different groups, IOPRI is reporting, and POB is doing that, and so on. So so many different sort of controls, but not there's no one actually can really say that I've solved the Ganoderma problem. And as I mentioned earlier, most managers or estates personnel, they fail to recognize the early stage of the infection. So 6 or 7 years, you see the symptoms come out. At that time, there's about 60 or 70% of the palm tissues are already decay. And at this time, when you see the symptoms, like what I showed you just now, is too late. Any treatment is not possible anymore because it's like cancer lah, like cancer, penyakit cancer. It already reached stage 4. So the problem that we have now is with this pump, the stage 1, stage 2, or stage 3, like penyakit cancer, they cannot detect it in the field. So this is the challenge. Okay, so what the planters are doing is still looking at the symptoms. For example, look at the first symptoms on your top left. So sometimes like this sort of skirting's a bit, they may not so sure whether is it infected or not infected. Okay, the one on the bottom and so on. And of course, if there's a fruiting body like the one on the right, yeah, and pointing it using an arrow, if you see fruiting body, then it's confirmed lah. But the only thing is the fruiting body does not come at that fast. Most of the time, when you see this fruiting body like this growing out, I think the fungus, which is the Ganoderma, have fit enough. Dah kenyang, full already. Then they start to disperse, ready to disperse the spore for next generation and so on. Then only you can see this fruiting body, right? So this is, for example, you look at the pumps. So this one actually, this is already severely infected in the middle here. Okay, so many different type of controls that are being done in the market. I mean, in the research, and is available. So people have been doing isolations. So they try to isolate the infected palm so that if you remember, I discussed about how this disease, how this pathogen spread. Okay, the root contact and so on. So if there is a palm infected, so what they try to do is they try to dig, like drain long parit. In Malay we call parit, or long drain. So 2 meters, and so on. So that actually this palm is being isolated so that it won't spread to other palm. And then many like soil mounding. Soil mounding is mainly because of some of the tissues is already decayed, so the palms can collapse anytime. And it's quite dangerous, especially if you go into estate that actually with heavy infection of Ganoderma when it's strong wind. So you can have one palm collapse in front of you and may just hit any one of you if you walk into a estate like that. So soil mounding is the one actually giving support, giving support to the palm so that this palm actually will last longer. Okay, will last longer, but we can still actually harvesting the yield from the palm. Yeah, so plowing and harrowing and all this stuff, sanitation and so on, planting legume crops, and some even crazy until perform surgery, removing of the infected materials, and then only do the soil mounding and so on. Yeah. Fertilizer inputs, so many research trying to play around with the so-called of increasing the magnesium, increasing the calcium, and so on, hoping that actually this type of elements and even boron, trying to improve the health of the palm so that the palm can be more can be healthier, or it's more difficult to be infected. Yeah, there are results here and there, but sometimes to some extent it's like is okay in this estate or in this trial, but it's not okay in another one. So mix so-called of uncertainties and so on. And chemicals treatments have been tried with a lot of different combination. Hexaconazole, azole group is the one, and maybe with different applications method also. Some even using pressure injection. So they inject it and pressurize because the infection starts from the roots, the roots which is in the soil. So what they're trying to do is actually they try to pressurize the chemicals into the lower part of the pump so that you can reach the target. And most of the time, I would say that chemical treatment is for Ganoderma so far. Chemical treatment is the one that reported with the least effectiveness. The minimum, the most not effective is chemical treatment. Quite interesting. For other diseases, chemical control usually is the one that actually give you a very fast control, and is usually reported the most effective. And to some extent, the only problem is maybe development of resistance and so on, or content pollution and so on. But for Ganoderma, chemical treatment so far is reported is the least effective. And people have been looking into biological control most of the time, but we'll share with you also biological control and of course some of the challenges that we can have. But to some extent, the most commonly practiced in Indonesia and also in Malaysia is this one, which is they will do a sanitation. Sanitation is very, very, you can see that how troublesome is it. So let's say this is the infected area. What they do is actually, for those infected palm, they need to, if you look at here, these are the the chips of the pump. So they need to chip it using this machine, okay, that you can see here, into smaller pieces, maybe about maybe 10 to 20 cm, chipping it into smaller pieces like this. And then, of course, the pump that infected, they need to dig it out, the stump inside the soil, and then chip it into smaller pieces like this, and expose it to the sun for some time, and before the new replanting can be done. So the cost is very high because they need to bring in a machine, and you know that the oil palm plantation is very big, so many and it's very, very laborious, labor intensive, and using, I mean, all these type of machines and so on. So, but unfortunately, this is the most commonly practiced in either in the big companies, small companies, smallholders, and so on. This is the best that they have been doing now. I mean, the idea is actually how clean you can make your plantation. That's only way. I mean, how good we can reduce the inoculum size to the minimum. Yeah, so we expose to the sun and so on. So this is the theory lah. If we look at the incidents also, we notice that if you have is interest in this area, you notice that the older plantation or older estates, the chances to have infection is I mean the percentage of infection is higher, incidence is higher. This is also, for example, like some of the estate in Malaysia, because in Malaysia we already reached 100 years of planting history. So an oil palm life cycles can last for maybe 20, 25 years before they start replanting. So meaning that 100 years, that will be about 3 going to the third, and some may going to the fourth replanting. So the third or the fourth generation. So imagine earlier, in the earlier stage where sanitation was not properly done. If you remember, I was discussing about this, how it's spread. So if the sanitation was not properly done, so some of these remaining materials in the soil accumulated there. So this is actually in correlation with the so-called of increase of incident, percentage of disease, when the cycles or the so-called of generation going from the second to third and fourth. Yeah. So because in the first generation, the sanitation was not properly done, so the accumulation of the inoculum is still there. All right. So getting more and more. Now I think they're trying to make it cleaner as much as possible. But sometimes after working with this disease for long, we notice that actually, do we really know our enemy? I mean, when we try to solve a problem, our enemy, we must really understand our enemy, the characteristics, how does it work, how does it spread, and so on. That actually that's only way we can control. Instead of control, I don't know Indonesian call what, but in Malaysia we call membabi buta, meaning that we don't know what is target, we just simply do whatever. So most of the time it may be not effective in that case. Okay, so interestingly, for those that not know Ganoderma, I think is okay. But for those that actually don't know Ganoderma, actually Ganoderma is a very poor competitor and weak saprophyte. We said that this is a facultative saprophyte, or maybe facultative parasite at the same time, meaning that they can live as a saprophytic, and if given an opportunity, they will become a pathogen. Okay, and it's very weak. Why is we saying it's very weak? Why is saying it's very poor competitors? Because it cannot compete with many microorganisms. That's why if we are talking about biocontrol, you can find some microorganism, and you put it on the petri dish, and it can easily overcome, and it can easily kill the Ganoderma. We are not excited at all, because it is very poor. The Ganoderma is very poor, it's very weak, and it cannot survive in non-sterile soil. This is what happened in the lab when you try to do it. And it be repeated in many researchers. The first one did it was the lab Dr. Richard Cooper in the University of Bath. And Richard Cooper happened to become he was my examiner when I did my PhD like 15 or 16 years ago. Yeah, so if you look at his research, very simple. 2 types of soil. One soil is sterile, one soil is non-sterile. Non-sterile soil meaning what? Means that they have some other microbes growing inside there. And we put in the Ganoderma inside there, so the Ganoderma is very difficult to grow. This is what you can see, the 4 days and 10 days here. All right, so only the non-sterile soil, only the sterile soil where the Ganoderma can grow. Okay, so basically this Ganoderma is very weak, is very poor competitor. It cannot compete with some of the microbes in the soil and so on. Yeah, so this is very interesting thing we need to know. Then it is also a very slow grower. Yeah, some of course they can have different isolate, different strain and so on, but some of them they if you put it on the petri dish, some of them even grow very slow. Some of them, of course, the more aggressive one will grow faster, but it's still much slower compared to your other fungal pathogens that you have been dealing with. I'm sure you have been dealing with other pathogens that grow much faster. Then also explain what we see in the field, because when we see the field, you notice that I told you just now, I shared with you, we took about 6 or 7 years to see the symptoms. So if we study the progressions of the infection, in fact, from the roots, and it progressed very slow. It's maybe 1 or 2 cm in 1 month time. So this is why you can see the symptom very slow. After a few years only you see the symptom. But when you see a symptom, it's too late. So this is the nature of the Ganoderma itself. But we are quite challenged why they are so nasty in the oil palm soil. I mean, yeah, if you try to do some artificial inoculation, you try to inoculate them in the nursery. So we need to have a big size of what they call this one, rubber wood block. We need to inoculate the Ganoderma on the wood block first before we actually can infect them. Okay, we can infect them. So meaning that they need a big amounts of inoculums before they can cause infections. Yeah. So this is the challenges that we have when come to dealing with this disease. But why in the field it can be so nasty? So I explain a little bit more when we discuss a little bit on the soil metagenomics. That actually we study to some extent we notice that maybe the applications of so much of agrochemicals, maybe the so-called of fertilizers and maybe the herbicide and so on, actually killing a lot of these beneficials or good microbes that actually can provide check and balance. And we believe this Ganoderma is actually very stubborn. It's slow, but it's stubborn, and it's grow quite. They're quite stubborn. This is why the other good beneficial microbes may be killed because of the normal agronomic practices, but this this Ganoderma remain there and growing slowly, steadily lah. Some of the management of Ganoderma using beneficial microbes that I will share with you. This is from our work in collaboration with Nestle. So Nestle, I think we have Nestle in Indonesia also. This is Nestle Malaysia. They sponsors the project under their CSR project. And then we are working with IOI and also Genting Plantations. A product that we tested here is what we call combinations between the Trichoderma and also Bacillus. So Trichoderma is the fungi, and then Bacillus is the bacteria that we use here lah. Okay, so in this case, what happened here is our experience is saying that sometimes it's quite difficult to work with a single microbe. I mean, single type of microbe. So from our observations after years, we noticed that most of the successful biological control agents actually comes from a group. I mean, it's like a combination, maybe like fungi and bacteria. So if they have a group, something that they will do different work. One will do this function and do another function and so on. So the application is very straightforward. This is an encapsulation technology where it's like come with like a pill. So once it's put inside the knapsack, water, it will disperse, and then we just spray it like that. Okay, so this is what happened. And this is in the field, and this is the one in the lab that we try to look at what actually happened in the field. So you can look at here. So this is the Ganoderma, the white in the color. Then this is actually the Trichoderma we are talking about, which is the Trichoderma bracteatum. So then we also have the Bacillus that going together here. So you can see that this is the basic. Usually when we do a screening, we go through 3 processes. The first is actually the basic, which is in vitro. The second one is we go to nursery, and the last one is actually we apply in the field, which is in the estate. So in this case, we are interested to know what actually this Trichoderma and all these Bacillus do when they meet the so-called of pathogen in this case. So when we look at here, so we try to scrape out this area when they first so-called of meet. So we notice that there's a lot of destructions actually caused by this trichoderma. You see these are images. These are the normal Ganoderma, so-called of the healthy Ganoderma without any treatment. But of course, these are the second, the third, and the fourth that you can just see here. Are those actually Ganoderma that are being challenged by the trichoderma and bacillus. You can notice that here it's a bit small, sorry. So the spores actually adhere at the myceliums of the Ganoderma, and after it grow, actually it coil the so-called of Ganoderma's mycelium. And interestingly, is the bacillus is producing antimicrobial compounds that's cause lysis to the so-called of mycelium of the Ganoderma that you can see here. So we screen through the so-called of what are the potential antibiotics or maybe the antimicrobial compounds that are being released by the bacillus. So in this case, we notice that, okay, this is the GB, we put it as a Ganoderma, and then this is the one that with the presence of the trichoderma and also TR1 is the trichoderma and also the bacillus. So because when we screen through, we need to look at some of the compounds that are being produced by the pathogen, for example, for their normal physiological activities or physiological functions and so on. And there are also compounds that are being produced by the trichoderma and also the bacillus, maybe for the normal life and so on. So we need to remove out those actually used for biological, for the normal physiological process. So these are the compound when they interact. Okay, so when there's a Ganoderma and mixed together, which is this one, the one I show you here. But of course, this is we're using Prof to do the study. So we perform under the chromatography system. So you notice that actually the so-called of bacillus produce these compounds that inhibiting the growth of, including this lovastatin, which is a very, very popular compounds that being reported for antimicrobial compounds activities, and others, including the dodecanoic acid also. Okay, this is also important. So meaning that the combination will work better. Then the next one is actually we try to look at the so-called of agostro content. This is the one actually we use to evaluate how effective is the treatment when we apply in the field. Just now I show you some photos here, which is the field. So this is the one that we spray out the so-called of trichoderma and bacillus to the field. So this is the in the so-called of in vitro in the lab, and when we spray through the field, this is the infected palm, for example. So next thing is we will take back the tissues to evaluate how effective is our treatment. Because after spraying it, if we want to evaluate based on the symptom, it's quite difficult because it will take quite some time to see the recovery and so on. So the quickest way is actually we use an indicator what we call agostro. Yeah. So if you're interested to look into more about this agostro, in human, like we have cholesterol. Then, but in fungi, they are agostro. So agostro is it's a wrong spelling. Agostro, ERGOS. So it's ERGOS, agostro. There's a typo here. Okay, so agostro is a fungi sterol. So in this case, but interestingly, it's quite labile. So meaning that once the fungi die, so meaning that this agostro will be actually degraded. So this can be used as an indicator of the presence of biomass, how much of biomass of this Ganoderma that present inside there. So if you look at here, so we have done so-called assessment. The color is gone missing. So the blue one is the so-called of before the agostro. Then the red one is the infection. So if we look at here, so these are the before treatment. So one, the control infected one. We also have the control infected. So we test on these two products. One is the LSM is the one that actually we have the Bacillus and Trichoderma. And then we have also an AO. This is another product which is using yeast as a biocontrol. I didn't show you the yeast just now because the result actually for me is not that promising. The results may still looks okay, but we cannot accept the so-called how the biological control is being prepared. I think the initial slide I did show you about the preparations of the biological control agents or biopesticide must be practical. Okay, for this AO, before it's being used, it must be fermented for 2 days first. Then you need to use a lot of requirements. The water must be clean, it must be no chlorine, and this and that. So, which is based on the feedback from the planters, the planters say this is not practical. So, I mean, nobody can have the time to do this type of thing. So that's why biological control agents or development of biopesticide also need to consider so-called of the practicability of the product in the field. Okay, when we look at here, notice this is 100% the infections percentage, so meaning that all the palms that we have tested, we screen through, collected tissues are infected. And what we try to do is also we try to have the so-called of agostro that are more or less the same at the beginning per gram of tissue. So meaning that there's no bias. So meaning that they have this more or less the same amounts of fungus sterol that present inside the tissues of the oil palm. Then only we treated it for about 1 year, 1 year to 1.5 years. We noticed that actually there's a drastically increase of those palm that actually not being treated with these biological control agents. And then this one we can see that they are going very low. Okay, going very low. So meaning that again, you can see that actually I won't say that this is an absolute, this is a totally 100% control, because you can see that the Ganoderma is not being killed, it's still there, but it's just so-called of the biomass, the amounts, or maybe we said the fungal count inside there much lower compared to those actually you don't treat it. That's why we have been tested a lot of products, even our own products and products from the market. Yeah, so we can't find actually a product that actually totally can eliminate the so-called of pathogens 100% and so on. But best as a recommendation as a consultant or as a scientist, if we are being asked whether we should, I mean the planters ask us whether we should be doing a treatment and so on, our recommendation is always look at the cost. All right, so how much is the cost of this product, for example, or this product, how practical is it, and so on. If they are investing $100 US, for example, then they're getting back $500 US. So that investment is worth, meaning that if they put it the product they just invested a small money, but they're getting more in return. But if they don't do any treatment, so meaning that the losses may be more. Okay, the infections may be more severe, and then it ends up with more. So these are the so-called of challenges they may face. So of course they need to look at the dollar and cent. If the control is not that expensive, they are getting more, then we are recommending them to do the treatment. And bear in mind, I did mention that until now we don't have any 100% so-called treatment that can work against this Ganoderma. We can only manage, or we can only reduce the so-called of diseases to the minimum. Yeah. So this is another one trial. We use another. This is other treatment also. You can see the before treatment and also after treatment. So most of the time you will see that there is no any 100% control, but we managed to reduce to the colonization of the Ganoderma based on the agostro fungal biomass to the very minimum. So which is to some extent is acceptable to some of the farmers or planters, because what they want is actually they want to prolong the life of the palm so that they can continue harvesting the fruits. Yeah, because most of you probably know that planting oil palm is a long-term investment. So they have been putting so much of investment money, everything in preparing the land, planting the trees, and most of the time they probably can get the yields maybe about after 30 months of planting. This is about 2 to 3 years time after planting. Then only they can start to so-called harvesting the fruit. Just imagine after 5 or 6 years or 6 or 7 years, then they notice that, oh, my palm is already infected. So the so-called of return of investment is still not being achieved yet. And to some extent, we will notice that once it's being infected, the palms, the so-called of palm, the number will be getting lesser, and the fruit size, sorry, not the palm get lesser, the fruits will getting lesser, and sometimes the fruits may be getting smaller also. Yeah, so these are the challenges. And the most challenging is actually the pumps collapse. We lost the pump stand. Yeah, so these are the things that to take into consideration. Okay, so the next one is these are some of the other examples that we have been working with all the different type of biological control agents, including actinomycetes. Yeah. So we are trying our actinomycetes also, and this actinomycete also was doing quite well. And then we actually sold this to a company actually. Instead of developing it on our own, yeah. So this actinomycetes actually is producing. Actually, it's a Streptomyces sundabacensis. So it's producing the same screening process that we undergo, which is using LCMS to identify one of the compounds that potentials to be released by this so-called of Streptomyces sundabacensis. So we noticed that actually it produces ribostomycin, benzyl malic acids, landomycin B, and also salinomycin. These are those actually contributing to the activity that we see in the in vitro. As I mentioned earlier, the screening process is very straightforward. We usually do three stages. The first is in vitro. In vitro is, as you see here, from the basic screening, putting on the petri dish and so on. So next thing is when we see the activity, it must be at least 80% of inhibitions and above. So the next stage is actually we want try to understand what contribute to this inhibition. Is it the 5 that we have been talking about just now, the mode of action? Is it just competition of space? Is it just competition of nutrient? Or is it producing antibiotics and so on? So when we want to investigate whether they are producing antibiotics or not, then we look into this, extracting the compounds that are being produced, and then to understand identify what are the compounds that are being produced during the interaction. And from there, we try to enhance them, meaning that whether the product is good enough to use as the microbe itself, or maybe we can actually produce the compound, the extract of the compounds, leaving the microbes behind. So we just apply the compounds. And we try to enhance by doing that. And then, of course, the second stage is the nursery that we were discussing earlier. Nursery. And then the last one is collaborations with the industry. If the industry is willing to collaborate, then because in the university we don't have big estates or plantation to be tested. So that's why the third stage, usually the third level, we need a collaboration from the industry. Yeah, and collaboration with the industry also very important, because there's also an endorsement. Yeah, because no matter how good is our product, if we don't collaborate with the industry, we don't get the so-called of rubber stamp, the so-called of endorsement from the industry. I found that it's quite difficult to promote the product, yeah, to be absorbed into the market and so on. Okay, this is still the same that I show you. Okay, we also have some others. For example, this is a Pseudomonas aeruginosa. This is also very sensitive actually. In Malaysia, we are now, I'm also involved in developing they just started actually. We do have it already, but we are trying to revise it back, which is under our Malaysian standard. We are looking into the biofertilizer now. The so-called of, because I believe in Indonesia, I'm not sure whether you have this type of problem or not. We have a lot of products coming into a country, sometimes claiming that this is the microbe they have, this is the cell count they have, CFU, this and that, and so on. But how actually we really want to control the standard of these particular microbes? Either is a microbe from outside, or is it microbe or biofertilizers that being developed from inside? This is, I think, at the very beginning of my lecture just now, I did mention about the so-called definition. Yeah. Sometimes some of them are biopesticide, but they claim as a biofertilizer. Some they claim as bioenhancer. And then a lot of definitions, this and that. This is sometimes the problem and challenges when it's not being monitored well and so on. So these are the challenges. So I think we are coming up with a standard to actually how to sort of evaluate this and how to endorse and so on. Okay, so, and talking about Pseudomonas aeruginosa also, there will be always a contradiction between plant scientists or agriculturists and maybe with those actually involved in health science, because we probably know that this Pseudomonas aeruginosa may cause problem to the lung, for example, to human being. Yeah. So how we can convince this the authority to use this Pseudomonas? It may be a different strain that not causing harmful to human. Yeah. So that's why it's very important to make sure that it must be we don't solve a problem, but we are creating another problem. So this is what they come to biofertilizer or biopesticide. Yeah. So these are some of the scanning images of the. This is the Pseudomonas just now. Pseudomonas. This is Pseudomonas, I think. Yeah. So how it's actually attacked the mycelium. So we always try to provide some evidence now. How actually the so-called of microbes work in terms of the five mechanisms that have been discussed earlier. Yeah, so because nowadays promoting a product to the industry, most of the time, especially for oil palm, is very challenging because most of the oil palm, those that have money to buy the product are those actually big company, and big company they usually have all their own agronomists, they even have their own scientists and so on. So they will need a lot of evidence how to so-called of how they work, mode of actions, and then how effective, and then they also need data and so on. Okay, the next one is this is the one that experience from Sumatra Utara. This is it happened that one of the students actually, Dedek Haryadi was my students working with what they call this one with oil palm disease, working with a company, and then did a PhD. I think he has completed his PhD quite some time ago, lah, a few years back. And this product actually already being used lah, as their in-house. I think Asian Agri have about, if I'm not mistaken, there are more than 100,000 hectare planted areas of oil palm. So they are actually producing in-house producing this trichoderma for their own use. I think it's good enough lah, because the area is so big. And of course, for the time being, I was made understand that they haven't so-called of use it for other plantation. I mean, outside of the company, but this is for in-house. So the screening started from, you can look at this. This is, I think, he has a few, like, these are trichoderma basically. The idea developed from isolating the trichodermas from some healthy palms. So through observation, it's quite interesting what we noticed at the beginning. There's so many palms actually infected by this Ganoderma. Then there's a few palms that are still standing there and look healthy. Okay, so then we suspect what actually hidden under the oil palm tree of this healthy palm. So because they are from the same varieties, the same batch of oil palm, everything is the same. They receive the same agronomic practices, the same fertilizer, everything. So what are the difference? Maybe from the soil itself. So they try to isolate, and we found we have a few trichoderma, which is name at the time is put under 523, 537, and 501. This is before identified. Then screening through the whole process, very normal. These are the normal in vitro screenings that most of the plant pathologists will do, looking at the activity, inhibitions, and so on. And then, of course, I did mention about characterization, which is we try to characterize, understand the potential biocontrol agents, and then we try to identify them. So after identification, it's actually a Trichoderma raceae. The 501 is the Trichoderma raceae. 523 is the Asperum, and 537 also is Asperum. And you look at, if you look at here, basically the raceae give a very, very good inhibitions. It's about 95%. Yeah. So we always try to look for the very high percentage, because for Ganoderma, it is so easy to control in vitro, I would say. Yeah, because we did mention just now that it is a very weak competitors and so on. So if we are talking about something less than 80%, we don't even look at it, because it's like nothing great, because this is a weak pathogen. So because when we screen through, we notice that the efficacies will drop. Yeah. So if you're getting 95% in the in vitro, if you drop through the nursery, and if you even drop more when we reach to the field, Okay, so this is good. 95, I think is the selected. So of course, undergo a lot of different process screenings, including antibiosis. Look at how actually it works, and then this is through the culture filtrate, and this is the SEM images. How it, this difference, this is the original Gano, and how it's actually react when come to the 501, 523, and 537. Okay, so you can see that's how it's being affected during this interaction. Further testing, of course, to the nursery. So in the incorporation of this trichoderma into the seedlings nursery, then we look into how actually this aspergillum and also Raceae, the three of them, Raceae, aspergillum, and also there are two aspergillum here, the three of them just now. They notice that this is until 10 months after treatment. You notice that actually, yeah, the control one is basically, this is the infected one, need about 83% already, but the Raceae is giving about 13.4. So this is about, so these two is not doing very well. Okay, aspergillum and also aspergillum 523 and aspergillum 537 is not very good, especially 523. Yeah, they are both aspergillum, but if you really look into the phylogenetic, they are actually slightly different. Okay, but Raceae is doing very well. This is about 13.4%. So meaning that they are still infected. Yeah, but it's just like the percentage of incidence will be lower and so on. Yeah, so in comparison to those actually control and so on. Control negative, of course, is not infected. It's a healthy pump. So this is the so-called of selection process. When we try to screen through the in vitro, if you remember, we are talking about 95% just now. And these two are 80%, but when it comes to nursery, this is very different, 71 and 48. And this is still maintaining quite good. And of course, after that, they proceed to the field. I mean, now they are doing it in the field. They're doing the so-called of upscaling and using it. And I visited Sumatra Utara actually in March this year under consultancy, but also for this trichoderma. We try to look into how actually to enhance their formulation. How much of the so-called starting amounts or concentration need to be incorporated. For the time being, they are using it for their nursery. So meaning that they treat it in the nursery, the pre and also the main nursery. Then after that, before they use this seedling well treated and then put into the field or in the estate. Now we are thinking about the possibility to so-called of apply that trichoderma into the field itself. So now meaning that they're using it in the nursery, treat it more for, I would say, preventive. So meaning that they try to protect, to try to enhance, try to do protect the oil palm seedlings before it's being planted into the so-called of dangerous field that have so much of this inoculum. But the next challenge now is we're talking about how about the matured palm in the field that is infected, how we're going to treat them. So this is why last March we went there, because I think most of you may have experienced discussing about trichoderma. This is a fungi. And you probably will know that actually trichoderma can grow at a quite shallow level of soil. So meaning that, and we need to understand where the Ganoderma grow in the soil. So actually how it works to the maximum. Yeah. Okay. Then besides looking on some other biological control agents to introduce in the soil, we also think about can the plant or can the plant defend itself? So just like human being, we do have our so-called of immune system and so on. So once we get infected by some pathogens, for example, some common flu, selesma, flu, then sometimes we don't take any medication, we can recover also. Of course, not COVID-19, I mean the common flu. So we can recover because our body fight against these pathogens by on its own and so on. So the same also apply to animals, the same also apply to plant also. Because if we get the ideas, the plant, if we encounter any danger, someone's bring a parang or a knife trying to attack us, we try to run away, to protect ourselves as much as possible. But for a plant or even like a palm here, it's been sending it there 24/7 or all the 365 days. The plants cannot move when there's any danger. So under the hot sun, under the rain, everything is must always be there. So being that then we of course we studies about plants defense and so on. We know that actually the plants have their own system how to develop. So we try to understand why the palms cannot defend itself from this Ganoderma at the very beginning. Then we try to screen through the so-called of all the different compounds that potential antimicrobial compounds from the palm roots itself. Because I was mentioning earlier that the infections start from the roots, the soil. So now we screen through the roots of the oil palm, and yeah, of course there are so many different types of compounds. But more interestingly, we found that there are 3 compounds that here I put it here that contribute to the resistant, I would say that, or maybe tolerance of that palm against the pathogen, which is syringic acid, caffeic acid, and also 4-hydroxybenzoic acid, 3,4-hydroxybenzoic acid. So these are the 3 compounds here. Okay, interestingly, we study, I think this is a very basic machine, I'm sure available in Undip also, which is the HPLC. So we do a time course study. So a time course study is meaning what? It's meaning that when we start to infect the palm, the roots, then we screen like 4 hours, 8 hours, 12 hours, and so on to look at the amounts of this compound. And interestingly, I would say that if we try to quantify it in terms of the amounts of these 3 compounds per gram of tissues, the amounts that present is only in microgram. I would say the range is about 70 to 110 microgram per gram of oil palm root tissues. Yeah. So okay, just remember the point is the amounts that presents inside the roots is about microgram. Yeah, microgram. So then we did an in vitro test. To kill this Ganoderma, we need the so-called of these 3 compounds in the quantity of milligram. Yeah. So then the compounds are already present in the oil palm roots naturally. All right. And then during the infection, once it's infected, these so-called of oil palm roots, they are trying to accumulate, trying to, just like ourselves, once we've been infected, so we are trying to develop this so-called antibody and try to get into a higher concentration of antibody. So this is also happened to the oil palm. So you try to go from microgram to milligram. Unfortunately, when we do the time course study, the 4 hours, 8 hours, 12 hours, and so on, we notice that there's a drastically increase of the concentration of these 3 compounds. Unfortunately, before we can reach to a concentration of milligram that can kill the Ganoderma, which is to kill the pathogens already has the ability to detoxify it or to degrade it. So meaning that to achieve the milligram that can kill the Ganoderma will never happen in the natural process. So this is how actually we come with the idea. This is another product actually, we call it as a formulations of phenolics. So this is the one that we see. If you look at here, this is the Ganoderma. And then if we increase it to about the lowest is about 0.5 milligram. So we be able to kill the Ganoderma without any growing. Of course, this is lower. So if anything lower than 0.5, they can still grow, but it's very, very slow and limited. But if you are 0.5 milligram, these combinations actually can kill them. So the idea to go back to this one is if we can reach a 0.5 milligram happening in the roots, actually it can solve the problem. But unfortunately, naturally, God created the oil palm only have in microgram of concentration of these 3 compounds. Yeah. So to go for this one, before it can achieve, it's already being degraded. Okay, this is being proven from the in vitro. So next thing what we do is instead of waiting for the palm to accumulate the compounds by itself, which has never happened through the process of metabolisms or degradations of the compounds by the pathogen, we formulate it the right concentration and then we apply it directly to the palm. Okay, so that's we help the palm. So of course, you can see 100% of control in the vitro. When we proceed further into nursery, we can still get a very good about 99% of control. But when we go to field, you can see that a very drastic drop, 90%, 60, only to 60%. Yeah, so the effectiveness. And of course, these are the challenges that we have faced. How effective is this product is being delivered to the target pathogen. From our experience, this pathogen actually grow most of the time, if you dig in the profile, the soil profile they live is about 30 cm or 60 cm. Anything deeper than 60 cm, you have a very little, small amounts of Ganoderma inside there. So what we did is last time we even dig a hole as deep as 30 cm and introduce that formulations directly there, so that the formulation can reach to the target as soon as possible. Because we must understand, in the soil there are so many other possible things that actually can lock the potentials of the bioformulation. There's also potentials that actually can be degraded throughout the process before reaching the target, before reaching the pathogens and so on. In comparison, compared to this in vitro and also to this nursery, this is a very, very straightforward effect. Yeah. So this is the one of the products of the formulation. With the name of Lestani, we call it Lestani. So this is we do a collaboration with Felda at that time, which is one of the, what do you call this one? MNC, multinational companies in oil palm company in Malaysia. So yeah, so besides pouring into the 15 cm or 30 cm of that depth of the soil, we also try to do this injection. All right, injection is being that we also inject into the so-called the trunk of the pump. So we drill a hole, okay, and then we use this we call it as a spring pressure injector. So once it's being released, so the formulation will go into deep as much as possible of the pump. Yeah. So this is a new packaging, and this is the old packaging of the Lestani. Basically, it's a formulation of fungicide contains phenolic acids. And we have done in vitro fungi toxicity, promising that we can see from the slide just now. And nursery trial, greenhouse trial, and then finally we go for the field. Yeah. This is what happened in the field after the treatment. Okay, so we can see that some of the symptoms here. So this is the visuals or disease symptoms before what do you call this one? Incorporation. This is tested in another plantation with the name of Chong plantations. This is with another one. This is the one, the Felda. Okay, and then this is another plantation. So we try to collaborate with different industry so that we can actually get the so-called of field data. So we have better combination of data. So you can see that these are the so-called of light skirting. You can see the broken fronds here, symptoms, and then this is more severe, the skirtings and broken fronds of the pump, and this is the fruiting body. Okay, this is a very near, crossroad of the fruiting bodies. Yeah. Then what happened is after the treatment, this is about 1 to 2 years of treatment, continuous treatment. We will notice that the palm with this is the palm with so-called no skirting or broken fronds after pruning. Of course, this we need to remove it because dead tissues or this sort of thing will never recover back again. But after you do the pruning is basically cutting down the fronds. So you can notice that actually there is no longer so-called of skirting or broken fronds after the treatment. And the next one is this one, which is we notice that interestingly, you can see the new roots. Okay, new roots growing out from the cavity area. So these are the cavity area. Cavity area is the one that actually you see the tissues already decay, and then it's being removed and so on. So meaning that there's a potential for them to regrow back. The roots are growing back. Then if we cut it deeper to see the internal tissues, this is the C. So you can see there's actually no more decaying in the dead tissue. So the decay is some sort of stop. So means that there's a very high potential that this product actually can limit the progressions of the disease. And of course, we still do the same assessment, which is looking at from where we get the 60% just now in the field, from 100 to 99 to 60%, we are still quantifying the amounts of ergostro. Okay, ergostro. So we noticed that without the treatment of this Lestani, the amounts of ergostro continuously to increase in a positive way. And these are the difference concentration. Yeah, we are looking at 0.4 gram of active ingredient, 0.8, 0.1.2, and so on. Of course, we try to look at the difference concentration, mainly because of the cost. Meaning that if we try to use a lower amounts of this active ingredient, we probably can get cheaper cost. So, but of course, the most significant one is about 1.6. I mean, the best when it comes to the higher concentration is better. But if we look at statistically, they are all the same alphabet, B, 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 B. So this ANOVA show is actually, with our bare eyes, naked eyes, we notice that they are significant. This is doing better. But if statistically, we can actually go as low as 0.4 because statistically it's not significant among these four. Okay, this is how actually we do the evaluations. Okay, same also apply to this also. These are some of the microscopic images. We want to understand how actually this product works, our formulation of phenolics after we have tested. So we noticed that this is how these myceliums of Ganoderma, this is the healthy one. Okay, they can grow happily, healthy, and so on. But with the presence of Lestani, this is the B1, which you can see that this is a 0.1 milligram, and this is the 0.5. You can see that's the differences. So how they're actually being disturbed. And besides looking at the myceliums of the Ganoderma, which is the pathogens, we also try to look at what happened to the oil palm tissues. So the one that we see here, for example. Okay, so what happened in the tissues after the application of this product? Yeah, but of course, this one, because of the monitoring process, we use seedlings to check so that we can actually monitor the progress. So this is the healthy oil palm tissues that actually not being infected by the pathogen, not infected by the Ganoderma. So it's look good and so on. But this one is the one that actually inoculated and infected, right? Then this one is the one that inoculated but we treated. So of course you cannot compare between the healthy and the infected one. But if you make a comparison here, at least you can see some recovery actually of the tissues that being treated with the Lestani. So this one is on the xylem bundles, and this is on the xylem vessels. So we suspect that this treatment, besides so-called of killing, or I would say maybe limiting and reducing the colonizations of the Ganoderma, it can also serve as a possibles of helping the palms to recover. Yeah, but of course we may need further investigation on this lah. Yeah, so far suppression is proven, and then the recovery may take some time. This is our product. This is another product actually. We have been testing it. This is not ours, but it's a combination of organic acids. Yeah, so you can see that the palm is this is a palm example, is severely infected. So you can see that I will say maybe about 50% of the basal tissues already cavity already decay and so on. And then until they need to support using what they call this one, the stick, so they can still standing there. So you can see floating body and everything. So, but interestingly, when this company came to us, they were claiming about this and that, and then so what happened is actually they're applying this product and so on. This is a combination of organic acids and so on. So when the palm is being cut off, I mean being chopped down, and when we notice that the palm that being treated with the product, they have a so-called of limited lesion. Because I think in plant pathology, we have a spreading lesion and we have limited lesion. Spreading lesion is mean that the disease is still active, the pathogen is still active. So it may be spreading and spreading. But this is for me more like limited lesions. Actually, it's like some necrotic spot that the infection may temporarily stop and so on. So is this just a observation? And what will happen next if we leave it for longer? Whether the disease will continue, but the colonization of pathogen will proceed and so on. So we evaluate the product itself, and we notice that inside there they have a combinations of all these different compounds. Propionic acid, acetic acid, benzoic acid, sorbic acid, also bacillic acid, right? So if you remember earlier, I was talking about the biopesticide can be many different style of category. Yeah? So not only limited to the microorganism itself only. It can be some plant extract. It can be some compounds and so on. For me, is most importantly is actually the effectiveness. How actually we can enhance the effectiveness, and then practicability. If it's practical, the microbe itself, just combination of microbe is good enough. You apply it, the effect is already there. So there's no point to look for other things, to look for to extract the compounds and so on. But if the compound itself can do better, and maybe the cost is lower or whatever, then maybe we can explore further. Same also, evaluation of these organic acids, we start with in vitro, of course, using different concentrations, 0.3, 0.4, and 0.5. And you notice that you can see from here, so this is the control, and then the different combination that actually stop the growth in vitro. And again, as I mentioned, if it's in vitro growth, so there's nothing so happy, because it may just so easy to control in vitro. And of course, we explore further what actually these compounds cause the Ganoderma mycelium. And it's quite interesting to see that this is the healthy one, the A one, control. And then these are different concentrations, 0.3, 0.4, and 0.5. You can see that's all this, the destructions of Ganoderma mycelium, erosion of cell walls, and so on in the different concentrations of treatment. When we apply in the field, the same also we evaluate the amounts of ergostrope. If you look at here, initially we will try to get the so-called of amount of ergostrope of pump more or less statistically not significant. So let's say if you have screened for a few hundred of pumps, you can notice that there are 108 pumps that fall into this category, 70 in this category, 91, 30, 16, and 10. So this is the amount of ergostro. Okay? Ergostro is the amount of fungal biomass. We use it as an indicator to test or to evaluate how effective is our treatment. Sometimes this is always questionable. A lot of people will say that how can you evaluate when it comes to Ganoderma? I'm talking about Ganoderma, sharing with you about Ganoderma. There's always challenges from agronomists and so on. How you want to evaluate your product is effective or not effective? There must be some indicator. If we want to look at a symptom, for Ganoderma and oil palm, I will say the symptom is very, very subjective. Very subjective. So it can, with no symptoms, but tomorrow the pumps collapse. No? Just like people's like no symptoms, suddenly tomorrow heart attack, die already. Okay, it can be pump with symptom, wilting and so on, but still standing there for quite some time before it collapses. That's why if you want to evaluate the effectiveness of a product or of an invention and so on by using symptoms is totally not reliable. I mean, for Ganoderma case. That's why we need to come with this ergostro. I would say that we are probably the first in using ergostro as the Ganoderma evaluation. Of course, Ganoderma, of course, this ergostro have been used in other plant diseases, but we were the first group. We are the first group actually to use ergostro for quantification. Although there may be still some argument saying that it is not specific for Ganoderma because other higher, all these higher fungi will have this ergostro. But I would say that the way in collecting the samples, actually we even sterile our drill bit before we drill in the pump and then removing the tissues and sterile again. So we have a standard protocol how the tissues must be collected so that actually we can evaluate the effectiveness of the product without any I mean, we can be able to accept the challenges and so on. So even when we started, when we screen through, we want the palm that actually, let's say when we're evaluating different product or even different concentrations of a product. So we want to make sure that the amounts of ergostro that the pump started to be treated is more or less the same, statistically not significant. For example, if we choose from this group, so the treatment A, for example, 0.4 shoot from this group also. 0.4, this is the difference round lah. This is apply for 5 round, meaning that lima kali. This is 0.4, 3 kali, 3 rounds, and this is 0.5 for 3 rounds. So, and then even like the previously I was talking about other product like LSM, AO, and so on. So we want to have a very fair comparison. So meaning that they are the pump and started, they are more or less the amounts of fungal biomass more or less the same. So meaning that at the end of the program or of the evaluation, 2 years or 3 years, or sometimes the fastest is 1.5 years. So we cannot say that the pump that let's say treatment A is doing better than treatment B. Then treatment A may be not happy. I mean, if the companies from treatment, they said, oh, maybe my pump started with a very bad foundation. It's already very severe and so on. That's why my product failed to manage or fail to control the disease. So we try to avoid this sort of thing. This actually we come up with some so-called of comparison. So started with more or less the same amounts of fungal biomass, so as a beginning. But of course, I think you may know that one is the pathogen, another one is the disease. So the disease itself maybe is a response from the so-called of physiological process. So how fast the disease we cannot control. Yeah, so we only can standardize, we can check the amounts of roughly indicators for the fungus itself. Yeah. Okay, but this is the way to make I would say the more equal or fair comparison, right? At the end of the day. Okay, let me just progress. Oops. We still have another 20 minutes, so try to go through. Okay, if you remember the 5 different mechanisms that we were discussing earlier, we were discussing about one of the things that we also think about the last one, which is how actually we can enhance. This is also one type of, we said a biopesticide also potential to be used. How we can enhance the mechanism, meaning that before the pump or before the plant or your crop is being infected, we can introduce something. So we're looking into chitosan. And we have tested on chitosan also. Why we look into chitosan? Because of the chitin. I think everyone know here that it's a long chain polymers of this all and so on. And we have so much of this chitins, I mean source, because in Sabah, I think Pak Buji came to Sabah before. So we were thinking about seafood. Come to Sabah is seafood. No? Makanan laut, we said. So we have all these of what they call this one, the shell from the crab, the shrimp, and so on. So actually we can turn this into something maybe useful because it's cheap. I mean, it's west and so on. So there's a potential because there's a similarity here. Because we must understand also the chitin is also one of the main component of the cell wall of fungi, right? So if we introduce the chitin earlier. In this case, so what happened here is the pump or the plant may sense the presence of the fungi. I mean, we mislead the pump, so the pump like terbangun, awakened because of they thought there's fungi coming because of the presence of this chitin. But actually the chitin may comes from your kerang or from your what do you call this one, crab shells and so on. So this is trying to boost up the immune system before the real pathogen can. Okay, this is just some definition. Chitosan is from how actually chitosan derived from the deacetylations of chitin. Yeah. So this is the one we have tested. Again, I use ergostro as the indicator because this is the one actually we can quantify. Because for, as I mentioned, symptom is quite difficult. Of course, we do observe from symptom also, but we need some quantitative data so we try to look into this potential of using chitosan as a preventive or as a curative. So preventive is mean that we introduce the chitosan earlier. All right. Oh, sorry, this is a different concentration. 0.5, 1%, and 1.5%. But of course, we do two. Beside the concentration, we also do. I think that my next slide will show you before and after. Meaning that is it the chitosan is good as a preventive? That actually we introduce the chitosan earlier to boost up the defense earlier, or we introduce it after the palm is infected. All right. So in terms of concentration, we notice that actually the higher concentration, you can see that this is the concentration higher is better. 1.5% is doing better compared to 1 and 0.5%. And of course, the untreated is yeah. So you can see what I mean. And this is the one I was mentioning about before and after. And we noticed that actually from our observation, it's actually better to what do you call this one? To introduce the chitosan earlier. Meaning that let's say if this palm is infected, I mean there are several surrounding palm infected. So for the healthy palm, you may try to introduce chitosan. Okay, so that's it will help to boost up the immune system of that palm, so that it may I would say prevent or maybe reduce the possible of infection. Yeah, so that's we can protect the palm. Okay, I think we're almost coming to the end. Another thing that I will still believe, if you recall back what I've been talking earlier, sorry for a long, so much of talking, but if you recall back, we are discussing about Ganoderma is a poor competitor. Ganoderma is a weak pathogen. It grow very slow. It cannot grow in non-sterile soil, and progression, infection very slow, and so on. So my questions are always challenging my students and sometimes my colleagues and so on. Why in the field it become like that? I mean, it's so nasty, in causing talking about 1 billion. I mean, in Indonesia, because Indonesia basically you have a larger plantation area compared to Malaysia, about almost double our plantation area. So this is the losses of 1 billion US dollar. In Malaysia, we're losing about 500 million US dollar a year, not collectively, but it's a year. So how can it be so nasty? We still believe that actually something happening in the soil. So this is the work that we have done. We look into the soil microbial communities. And of course, I'm not going to go into very details about this research, but what I'm trying to look at here is actually we noticed that these are some of the data that were collected from the Ladang Sahabat, which is the Velda MNC group I was talking about just now. We noticed that actually from here, what I try to conclude for this work is the soil condition or the soil health, I would say, is very, very much affected by the agronomic practices. We screened through a few different so-called estates. One is a big company. Probably I don't want to mention a company name now. The big company. The second one is I would say SME, is small medium enterprise, and the last one, another is a orang kampung style, which is a very, very traditional, small scale and so on. So they have very big difference when it comes to agronomic practices. Of course, everyone is still putting fertilizer, doing basics weeding and so on, potong rumput, rumpai and everything. But the difference is those they're using biological control agents, and then using organic materials and so on. We notice that the diversities of the microbial community is much better. And then we also screen through the so-called of different incident, the high incidence, the low incident, and then the healthy palms area. And we noticed that you can see that high BSR disease appear to affect soil communities also, meaning that after some time, this Ganoderma maybe can dominate and suppress other microbes that growing there. And then the presence of Ganoderma also has impact on the community structures. Yeah. And the one I was talking about just now is the different microbiome communities could be corrected with field management. What we see is actually those actually very badly managed are actually those big companies sometimes to some extent. The pH can drop very, very low. The pH of the soil can drop very low. And then the so-called diversity of the microbes inside there can be very, very quite bad. We didn't find many good those microbes actually can do the nitrogen fixation. They can enhance the defense mechanism and involved in phosphate solubilizations and so on. So these are maybe one of the reason, and it's very difficult, to convince. I'm sure that some of you maybe have experience, especially working with those big company, because they have like a standard protocol. Meaning that after how many months they need to put in the fertilizer, after how many months they need to do this and that. If you are talking to some small scale farmers and planters, it's much easier to convince, because probably they are the boss. They can take the decision and so on. But when we try to talk with big company, they will say that if you want to reduce this and reduce that, if any drop of yield, are you going to pay us back? So this is then we need to come with data and everything. Even I remember we have collaborated with one very big company doing a testing for about 3 years, where we reduced the amounts of fertilizer to maximum of 50%. Yeah. So we reduced 15, 25, and 50. So 3 different sort of treatments. And of course, we replaced with some organic materials and some of these microbes and so on. So the first questions from the company that time was asking us is, are you going to take responsibility if my yield drop? Because we know oil palm is like a golden crop, so much of money, everything. So they said, I'm going to lose my job, if my yield drop and so on. Are you going to take responsibility to pay me back if my yield drop? Then we discuss and so on, and finally managed to convince, to proceed with that assessment for 3 years. And interestingly, after 3 years, even with the highest reductions of fertilizers, up to 50%, the yield has never dropped. Yeah, it still maintained the same yield. So what is the things we're talking about is actually we can save a lot of money from the fertilizer. That's the first thing, and we can make the soil healthier and so on. Now the second question come. The managers, it's really a director, told us that, oh no lah, my soil 3 years is not enough for you to get the data. You need to assess it maybe 5 or 6 years. So because my soil is still with a lot of nutrient inside there from my previous fertilizer. So you see, sometimes it's so difficult to convince people. They said the first, even we want to reduce 15% or 25%, they are so noisy already. If you're talking about 50%, if the yield drop, are you going to replace me? So I mean that they are very confident that not putting fertilizer in is going to give a yield drop. But now, when we prove it after 3 years, no yield drops and so on, then they say it's not enough, not long enough. 3 years is not enough. You need 4 years or 5. I think he was talking about 5 or 6 years. Then you see, these are the challenges when it comes to research and dealing with the industry and so on. Yeah. But soil bacterial abundance and diversity is also vary substantially under different soil physical, chemical properties and so on. So these are the things that are very interesting. And this is probably my last slide. It's not really on Ganoderma. This is one of the research that we just collaborate recently with a group of researchers from China and also from Sweden. But the professor just moved to Netherlands recently. This one we are using the so-called of AMF mycorrhizae, which is a Phonolophomis mosaic. Okay, this is the mycorrhizas fungus. So we use this Phonolophomis mosaic to do few things. One is actually we are trying, because the soil, the black soil in China, they have this problem, actually heavily polluted by antibiotics. And these antibiotics, of course, it can come from healthcare, can come from the livestock industry also, our cattle, our chicken or whatever, so feed with antibiotics and so on. So then the secretion, kinjak, kumbahan, whatever, come out from the animals, it's going to pollute the soil. And the black soil is very important for them. This Chao Yang is my PhD students. So what we try to do is to use this Phonolophomis, trying to whether this AMF actually can do some degradation of the antibiotics and trying to revise back the quality of the soil. And of course, in this case, we are using soybean, as an example. So I share with you this one because it's one of the latest paper we just published, I think like last month. It's a very good journal on this one. So so what we have proven here is you can check this paper if you like. I think it's open access, yeah. So you can get it free of charge. I mean, you download the full paper. Interestingly, this AMF actually not only can degrade the antibiotics, but when we use soybean as the model, it can also improve the soybean, improve the soil microbes, the soil metabolites, and so on. Yeah, so I think I put up some words here also. Total nitrogen content increase, and then all these sort of things. So I believe that it is very interesting. As long as we still love this biocontrol, biopesticide, and so on. Okay, so now this is the last part. The lessons that we learned here, the last part of my talk today, I will summarize the 4. The first is combinations. Combining multiple microbial strains may be more effective as biopesticide against plant pathogen. I think I mentioned this earlier because we have been testing so many one type of Ganoderma or maybe one type of bacteria and so on. And we noticed that actually most of the time it's quite difficult. It's quite difficult to be effective. So most of the time, of course, not saying that not effective, it's still effective. But combination, if you can find a combination, then when doing combination, there's another challenge because we want to make sure that we screen through a compatibility test. So meaning that they must be compatible. We don't want the two biological control agents fighting against themselves before we formulate them and upscale it to a industrial scale or whatever. That's the first thing. The second thing is we learn also understanding plant pathogens and their environmental interaction is essential for effective control. We really need to understand like this Ganoderma also after we're doing this again and again, this and that. I say why not we go and really study these pathogens and understand them better so that we can control them better. Most interestingly is actually there's another more crazy research that we have been doing earlier is actually I even keep controlling this Ganoderma. At the end of the day, I was thinking about why not we as a plant pathologist, people are questioning me also. But I was thinking about why not can we use this Ganoderma for our human health and so on. Okay, yeah, just now we are talking about Ganoderma lucidium, the lingzi, which is the Chinese community used for health supplement and so on. And this is a Ganoderma boniniense. Different species are the same genus, but the effect can be very different. We evaluated actually, lingzi, the Ganoderma lucidium can be used as a supplement. Supplement is mean that when we take it, it boosts up our resistance. Boosts up our immune system. We are getting healthier and so on. But Ganoderma boniniense, when we screen through, we undergo a long process also screen through. We look into the compounds and we have tested until using rats, using ticus, the rats tested and so on. And this Ganoderma boniniense extract actually has a few compounds which is very, very good in killing methicillin-resistant Staphylococcus aureus. Maksudnya, killing the antibiotic resistant. I mean, those pathogens that causing disease to human, they're actually resistant to antibiotics can be killed by Ganoderma boniniense extract. Yeah, mainly because of agrosol is one of the compounds, lanostan, and also the other one I can't remember. 3 compounds that we found from there, and it is quite safe to be taken. I think not mistaken, if I can remember, it's about 800 to about 1,000 milligram per kilogram of body weight. So meaning is that this Ganoderma boniniense maybe can be developed as a drug, not as a supplement. So meaning that we use it to kill when there's an infection or whatever, we can take it to kill the pathogen. But Ganoderma lucidium, the lingzi, we take it every day as a supplement. So there's just a different species. So that's why talking about understanding just now, I don't put into this slide just now because we are talking about biocontrol. Yeah, so until like sometimes we feel like we need to understand this pathogen so that we can work better. Yeah. The third one, integrated crop disease and pest management offers a more sustainable long-term solution. Yeah. So for Ganoderma, for the time being, I would say that there's no any single control. So most of the time, like the one we have been working with Sumatra Utara also, they have integrated. So sanitation, I mean, they have a set of protocols. If the palm, I mean, if that area, how many % of incident they may do treatment. If there's no, if more than how many %, sometimes they even just leave it like that. So meaning that it's not economically wise to do any treatment anymore. So, even they will probably it's very severe, they will even stop fertilizer. no fertilizer, no treatment. Yeah. And then what they do is they just collecting the fruit. Collecting a yield until certain when the palms collapse to certain percentage, then they will do replanting. So this is why we call it integrated lah. So different come with different what correspond plan. Treatment first, treatment can come with different type of treatments and so on. So like even detection also, like the Japanese company come, you fly on the foliar, look at the foliar, the top, then our team will go into to use the scanning differently. Okay, the last one is I still believe the soil health is extremely important. Yeah, and plant health. This is a theory. I think it's not something new, but it's something that we must champion. This is why I always, when I talk to the industry, we always try to educate. Not say they're not educated, but it's just that sometimes they may follow because the oil palm history have been so long, 100 years for Malaysia, for example. It's so difficult for people to accept new changes and so on. Yeah, so but regenerative agriculture with a focus on soil and plant health could be the way forward. By looking at understanding that the Ganoderma actually is so weak, but how it can become a strong enemy just because actually we are the one providing the so-called condition for these pathogens to excel. Okay, thank you very much for your attention. I'm so sorry I spent about really 2 hours. So this is my email address and my number. If you, of course, I can pick up questions after this, but if there's anything I cannot unable to answer or don't have time not sufficient, you can always text to me also. Back to you, Pak Irfan. Thank you so much.
Okay, thank you so much, Prof. Chong, for your comprehensive and also interesting presentation. It's very interesting to ours, I think. No problem, Prof, because the topic is very interesting, and I also have so many insight from the presentation. Okay, now we will move into the Q&A session, and then maybe I will read the question. Maybe we will take a Q&A session 10 until 15 minutes, Prof. Okay, maybe I will read the question from Luis Devina. Excuse me, Prof. My name is Luis. May I ask if the use of Lestani or other biopesticide product as a solution to Ganoderma attack can be done even before there are a sign of infection, or do we need to know first that the plant is infected? Jadi, apakah bisa digunakan terlebih dahulu atau harus tahu dulu, Prof, bahwa tanamannya sudah terinfeksi? Okay, please, you can answer it from Luis Devina.
Okay, this is about. I think I found the questions also here. Sorry, the voice is not very clear. So okay, the use of Lestani
If you
before
want
any
to,
signs, or do we need to?
can we use the biopesticide to the healthy plant, or we must know if the plant is infected? Jadi we must know the plant is infected, and then we apply the biopesticide, or we can apply it before there is a sign of infected.
Yeah, understand. Thank you, Pak Irfan, and also Miss Lois for your questions. I would say no, because we don't apply Lestanis when the palm is because just now we were discussing about we can do preventive. For example, we apply chitosan and some others microbes, especially microbe-based bioformulation, but not for Lestani. Lestani is phenolic acids. It is more for curative purpose. I mean, it's more on treatment. Means that we usually apply it when it is being infected. In this sharing just now about 2 hours, I didn't share about our research on early detection, because 2 hours is too short to share about the early detection also. Because for me, the Ganoderma control, it must come with an integrated, meaning that there must be an early detection and there must be a control. So these are the two challenges that we face in the oil palm industry when it comes to Ganoderma. Early detection is meaning that we must detect the infection as early as possible. The in situ or in planta, in the field, meaning that we must be able to identified the palm. This is why we have come up with a Gano scan that actually we use acoustic wave to generate the images. We mean that we can like scanning the baby now, scanning the baby in for those medical doctors. So, but even this one, we can scan the palm and generate the image to see where is the location of the cavity or the location of the infection. Then we inject into it. So that's why Lestani, we only do it for infected palm. We don't do it for preventive. For the time being. Yeah. I hope I answer your questions.
Okay, it is clear, Luis? Or you want to give a feedback?
Yeah, maybe I can just read from here lah, kan?
Oh yeah, got it. Thank you. Okay.
So the second question is from
Nurfianto.
Nurfianto.
Yeah.
I hope I pronounced your name correctly. It was mentioned in the presentation that Ganoderma can't thrive in non-sterile soil. Does this mean that intensive agronomic and oil palm monocultures render the soil to be sterile? Then why do agronomic practices can compost from empty oil palm bunches, compost from chopped oil palm trunks that have been cut down have no significant effect on reducing disease incidence in the field? Okay, maybe I go one by one now. The first question is on the sterile issue. Whether I won't say that we sterile the soil by doing all these agronomic practices, but it's just like something I would say mimic. Like if we try to look at the scenario that they cannot grow in a non-sterile soil. And of course, in the laboratory, it's always very difficult to start the infection. So there's a challenge. So that's why we try to use something sterile and so on. But in the field, of course, the agro practices give impact to the. I think when we talk about the soil metagenomics, there was one slide I show about the diversity of the microbes inside it. We noticed that the agronomic practices, especially the usage of extensive agrochemicals, actually do harm some of the important beneficial microbes and so on the soil. It is not sterile. I mean, the soil in the oil palm plantation, but this is not possible sterile. But it's just like the good microbes that's supposed to provide a check and balance to these pathogens may not present there anymore. That's why it very depends on the control now. The second question is talking about applying all these organic materials, maybe I would say compost and so on, have no significant effects on reducing disease incidence in the field. We must understand this one thing. It's like whether we are providing some support to the palm so that the palm can go healthier, or we are providing some support to the soil so that the soil can be enriched back, so that some good microbes can be coming back and so on. That's the one point. The second point is whether we are providing some direct control, which is attacking the pathogen itself. This is the next thing. So for me, all these oil palm bunches, compost, and all this sort of thing is actually not a direct control against this pathogen. It is more like we are enriching the soil and making the soil a better place for this good microbe to stay and so on. And bear in mind that this sort of thing may take some time to be seen. And most of the time when we do our evaluation, be 1 year, 2 years, and so on, we may unable to see the effect very directly, especially in the field. But if we use the poly bag, we do it in the nursery, probably we can see the so-called distribution of microbes and so on much faster. Yeah. So I hope I answered your questions. Then you have another one. Biofungicide products presented in the presentation effective against various strains of Ganoderma that as far as I know, I can mutate. Yeah, interesting questions. This Ganoderma, they change quite rapidly. Even from our experience, if you keep maintaining in the lab, after you subculture for quite some time, it may start to change also. This is possible also. I won't say that it's effective for everything, but the one that we have tested so far, it is effective lah. But effective again is not 100% control. No? I think I did show you the figures, everything, and again, even when we do consultancy, also either with our own products or with the products from for us is we always open to collaborations. We open to even to the industry. They come with products and so on. I mean, for us, it's like as a scientist, we are looking for solutions for industry. Of course, if it is something from Undip or from UMS, or from ourselves, it's something better lah. But if it's from the industry, from a company, I think it's okay also. We just want the solution for industry. So that's why we always come to this evaluation. Then the recommendation is always, like I mentioned earlier, it's like, look, we need to check back the so-called the cost. If you are getting $500 back instead of spending $100, for example, so it may be worth. So because after you minus back the cost, you still get another extra of 400. Yeah, but if you don't spend the $100, so you probably lost more of it. You lost 1,000. So I mean, at the end of the day, our recommendation is always whether you mampu or not, whether you afford or not, the company afford or not. If afford, they're getting more from what they invested, is worth to do lah. Because it's not an absolute control, but at least it can reduce, can suppress, and they can prolong the life, and they can still harvesting their fruits lah for the economy. So
Okay, thank you for
okay,
the answer.
yeah.
Okay, is there
I
any
think no more
question?
questions.
Okay,
Any more
thank
questions?
you.
No. Yeah.
Okay,
Any more questions there?
okay, what?
Did I miss any questions?
Oh,
I think
there is any
only two
question?
questions, right?
Yeah, only two question. There are any question from the participant? You can raise your hand maybe if you want to interact with Prof. Chong. That's not the problem.
I hope I
Or
don't
not.
speak too fast, but if I...
No. I think we can we can get the pace. It's very clear. I think.
Thank you.
Okay,
I hope
thank you.
we can I hope it's benefiting, and we can collaborate maybe. Most of them are students, or is it
Yeah,
some
some of this is some of our student, and also there are several lecturer
I
in this
see.
forum. Okay,
Yeah.
if there are no question, I want to ask one question maybe for Prof. Chong. The
Yes,
last
please.
question. Okay, maybe because my background is microbiology and I like to explore the bacteria. And then I want to ask one question is that you say before that when we use the bacteria as biopesticide, we must choose the bacteria that have a genetically stable. It has a stable in genetic, yeah. Because in my experience too, when we reculture the bacteria, its ability is decrease along the time. So I want to ask, when we get the bacteria that have a great potential even for biopesticide or another things, how can we keep the bacteria have a stable genetic compound so it's not have a decrease activity from when we isolate it? Yeah, maybe you get the question, right?
Yeah.
Okay.
Thank you, Irfan, for the questions.
Okay.
Right. Maybe I ask another questions back to you. Is how after how many cultures actually you have this type of problem?
Maybe 4 or 5. When I isolate the culture, actually maybe I reculture it 4 times before I use it to produce something. But when for example, when in my undergraduate studies, I search the bacteria that can produce indole acetic acid from the rice field in Indonesia. And when the first time I test it, it's a very high. It can produce the high value of indole acetic acid. But when I want to test it in the next month, maybe 2 or 3 culture after that, the production is very decreased. So how we can keep the stability production and in the microbes that we obtain, because it's very important to scale up the
Yeah.
bacteria.
Understand. Yeah, if you look at what we have discussing earlier, is actually we are thinking about how actually we can enhance I mean, besides the activity itself, we also try to enhance it by doing some extractions, if you remember what we were discussing earlier. So sometimes instead of looking at the biological control agents or the microbe itself to perform, we may look into what are the potential. This is another option now, besides using the bacteria itself. Is what are the compounds that actually contribute to the activity that you want. So if we were able to do, because okay, for the development of biopesticide, there are several different level or stages. One of the highest one actually we are thinking about, which is synthesizing the compound, which is actually that one will involve the chemist. I think most of the time we as a plant pathologists or microbiologists, we won't be able to do that. But so if we be able to identify what are the compounds that present inside there, so instead of relying directly on the bacteria itself, we might even think about how actually we can. One is to extract it out. The second one is actually if the compound is easy to be synthesized, we can actually have an organic chemist to synthesize it. So we may come out with some things. Maybe it's no longer a biopesticide, but if it can be something very potent. So this is the longest one. But if the bacteria is so easy to upscale, it's so easy to, is so low cost and so on. So there's no point actually for us to extract and then to, to get this sort of thing. So, but for your case, if it's really challenging to maintain the so-called of stability and so on, I think Pak Irfan what I think about gating the compounds that may contribute to that instead of relying on the bacteria itself. But if the bacteria is very stable, it's so easy to maintain and so on, there's no point actually for us to go until that level to synthesize it or to extract it and so on. Yeah. But if your micro is very good and then your compound is very good, so why not we look into a higher level, which is actually how to synthesize the product and upscale it without fully relying on the bacteria itself. Yeah.
You
I hope I answer your
recommend,
questions.
yeah. So you recommended to extract the compound rather than using the bacteria.
Yeah, either you extract the compound or maybe you can after you study the structures of the compound, we can actually synthesize it. You need an organic chemist to do that. So that will actually guarantee the pipeline productions. Usually for biocontrol, we look at different angle, the cost, the practicability, and then something like that. Yeah. So don't. I mean, usually we don't stop with one angle only. So we look at which one actually is more practical. I always been telling my collaborators, I said we always try to look at which is practical, cost effective, and so on. Don't stick with something that we know only. Yeah. I hope I answer your questions.
Okay, yeah, it is very answer my question. Okay, thank you. Actually, I want to discuss more with you, but the time is up. So maybe thank you very much, Prof. Chong, for the interesting presentation and also discussion. And all of you, if you want to discuss more about this topic or another topic, right? You can
Yeah,
we
sure.
can
Thank you so
contact
much.
you. We can contact you by the
Sure.
number or the email.
Yeah, you can send me an email
Okay.
also. Yeah,
Okay,
thank you so
thank
much
you.
for your time. And then I really hope
Okay.
the students pick up something. And for
Yeah.
those lecturers and faculty, maybe if there's a chance to collaborate, and
Yeah.
welcome to our university also. Yeah.
Okay.
Thank you so much.
Okay, thank you
Have
so
a nice
much, Prof.
evening.
Chong. Okay.
Bye-bye.
Okay, maybe I will give back to the Master of Ceremony.
Oh, okay.
Thank you.
Okay, thank you very much to Prof. Chong for the insightful presentation. And I also want to thank to our moderator, Mr. Ivan, to leading us today in this event. And now we will proceed with the certificate presentation to our honorable speaker. And I would like to invite Dr. Sri Pujianto to please joining us to present the certificate.
Sertifikatnya?
Uh, ada.
Ya. Belum muncul, Pak.
Yeah, it's due to slow connection in here. Okay. Okay. We have a problem, Mr. Puji.
Kok belum ada?
The connection is not stable in here, so maybe you can give a speech. No. Nah, ya.
Nah,
Sip.
oke. Terima kasih, Prof. Chong. Ini sebagai tanda
Thank you.
terima kasih kami sertifikat sebagai speaker pada kegiatan visiting professor.
Yeah, thank you so much, Pak Buji. Hope to see you again in UMS. Yeah.
Okay, maybe we would take a picture for a second, maybe, and I will count. Okay, the operator. Okay, I will count. 1, 2, 3. once again. Okay, 1, 2, 3. Okay, thank you to Prof. Chong for joining us today.
Thank you. Bye-bye.
Oke, terima kasih Prof. Chong. Ya,
Jumpa
sampai
lagi.
ketemu kembali.
Thank you
Okay,
for the
thank you.
thank you for the
And we kindly remind all the participants to fill out the attendance form through the link in the chat box. And ladies and gentlemen, we have now come to the end of the Visiting Professor online lecture on biopesticide and beneficial microbe for sustainable crop protection lesson from Ganoderma. And on behalf of the committee, I would like to express our deepest gratitude to our speaker, Prof. Chong Kim, and our moderator, Mr. Irfan Anwar Fauzan, and to all the participants for joining us today. And we hope this lecture has been meaningful and beneficial for all of us. Thank you very much. Stay healthy and see you at our next event. Good afternoon.